Hello everyone. In this module, you will see the discovery of the cytokinins. During 1940 and 50s, an intensive research was carried out on plant tissue culture. The plant tissue culture still in a primary stage. Using plant tissue cultures, it was possible to keep cells alive but difficult to induce the growth and cell division. While studying the plant tissue cultures, it was discovered that addition of the coconut milk induced the growth and cell division. That means coconut is an ingredient which supports the growth and cell division. Different group of scientists made a similar observation with the extraction of the yeast and denatured DNA. All denatured DNA promotes the cell division and tissue culture. That means denatured sample of the DNA had something which stimulates the cell division. The new compound was modified form of adenine termed as kinetin. Kinetin is a form of cytokinin. Cyto means cytoplasm. Kinesis means division. So it becomes cytokinin. Cytokinin is the substance which is believed to enhance the process of the cytokinesis, that cytoplasmic division during the cell division. Cytokinin or kinetin. Haberland in 1930 observed that the vascular sap is capable of inducing the cell division in wounded potato tubers. Similar capacity was observed by Van Overbeek in 1941 in coconut milk, which is a liquid endosperm. In 1954, Miller isolated an active substance from the autoclaved DNA of erring sperms, which was stimulated the cell division. He named that substance as kinetin. It was identified as a 6-perpyryl aminopurine. It is modified form of adenine, a purine. Kinetin does not occur naturally in plants. Several names were given to this substance, such as kinetinoid by Burstam in 1961, phytokinin by Osborne in 1963, cytokinin by Letham in 1963, and so on. However, the name cytokinin preferred over other names because cyto means cytoplasm, kinin means division, and abbreviated as a CK. Skogan et al. In 1965 defined as a chemical which, regardless their activities, promotes cytokinesis in various plant parts. The most common physiologically active cytokinin is zeatin, dihydrozeatin, and isopentenyl benzyl adenine is regarded as a synthetic cytokinin. But Ernest in 1883 found 6-benzyl adenine riboside in the seeds of Pimpinella anisum. Skogan and his co-workers discovered these regulators Cytokinins are synthesized the area where cell division occurs. Example, root apices, developing shoot buds, and young fruits. Petandas and Skoog reported that kinetin promotes the cell division by activating the DNA synthesis. In tobacco pith, it has antagonist property with auxin. Kinetin promotes the lateral buds, neutralizing the effect of auxin. In 1960, Thimann reported that the kinetin activates the protein synthesis in birds by incorporation labeled amino acids. Owing to this ability to bring about the cytokinins in cells, the name kinetin was given. The presence of kinetin was reported in extract of coconut milk, endosperm of maize, and tender apples. In 1956, Aurora and et al. Reported that the cortical cells of tobacco root enlarged. In 1963, Letham isolated another type of cytokinin is zeatin fractionation of sweet corn. The high activity of zeatin is due to side chain of very reactive allylic hydroxyl group. According to Letham, in 1974, the zeatin and zeatin ribosyl zeatin are most reactive compounds. The higher activity of zeatin is perhaps due to a very reactive allylic OH group in its side chain.